Okay, so we're very happy to see everyone back this morning, and also to welcome Professor Bill Newsome, who's from Stanford University. Bill is the professor of neurobiology at Stanford and is active in the whole area of cognition, and particularly in the context of vision, has written a huge number of publications in that whole research area. As a member of the Academy of Sciences in the United States, that's the equivalent of the Royal Society in this country. And you'll see rest of details on your biography there in your handouts, but there's also one whole list of various kinds of prizes, which I won't try and read out now. So we're very pleased that Bill has come to us to speak, and he's going to open up this whole area of the neurosciences, faith in a major science, the challenge of the neurosciences. Thanks, Bill.
And so I'll be talking today about my perspective on sort of some issues that are fundamental to Christian faith in an era where neuroscience is likely to be a dominant science over the next century. And I'm told that I should always start a lecture with a big picture, telling you this is the slide that John Bryant alluded to the other evening. This is the big picture. It's a big picture indeed. And neuroscientists, of course, think that this part of space inside your cranium is just as interesting as this part of space. And indeed, the brain, this 3 pounds of collection of cells within your cranium, is what you use to accomplish all of your interactions with the world, both appreciating the world and acting on the world. And in the old days, many of you know this diagram here comes from a movement called phrenology in the middle of the 19th century, where people analyzed brain function by feeling the bumps on your skull. So you would go into a phrenologist, and people would carefully palpate your skull, and depending on where your bumps are, they would identify, that you could be a very secretive person, that there was a center for cautiousness, and there was a center for appetites, and there was a center for, I think they're a little blurry, I can't read them very well, but you can basically have your mental faculties probed by feeling the bumps on your skull. Now, that's not the way we do things any longer. We know now that this brain is composed of about 10 to the 10th or so neurons, and most biologists, what's an order of magnitude among friends, right? About 10 billion or so neurons, and these neurons interact with each other through electrical impulses, very brief electrical impulses that last about 1 millisecond, and this is the currency, the coin of the realm, the currency of trade inside the brain. And we tend now to analyze various processes inside the brain that would include sensation, how energy in the world gets transduced into these electrical impulses in your sense organs, such as the eye. But the eye, the activity of the eye doesn't completely determine exactly what happens in the central nervous system, because we have added constructs inside the central nervous system related to perception. And many of you have probably seen this very famous little illusion here that it connects a triangle, and you see this black triangle standing out very clearly, even though there are no boundaries right here. And what this means is that we organize our percepts inside the brain in ways that organize in ways that are not completely determined by the retina here. And there are decision processes higher in the brain, and I've done a lot of work over the last few years studying how decisions get accomplished in the brain. And after we make decisions about what's out there in the environment, there's typically some motor planning that goes on. We may plan a trajectory of arm movement that's some sort of velocity function as a function of distance here, and ultimately we act on the world. So there are movements that get made and that accomplish our interactions with the world. And within this entire cycle here of sensation to movement, there are various internal mental processes that psychologists and neurobiologists are interested in, such as memory, attention, motivation, and emotion. And these are all very active areas of research, trying to understand what exactly are the physiological mechanisms underlying these various mental processes. And the notion in the neurosciences is that all of our behavior and all of our mental abilities are somehow related to the electrical activity of these nerve cells in the brain, and we're trying to understand what that relation is, what the mechanisms are that underlie our mental lives, if you will. The notion is that you take away the brain and you take away the mental life. So there have to be some sorts of biological mechanisms, and those of us who are interested in this sort of thing believe that this will actually be the most active and exciting area of science in the 21st century. Much as molecular biology was in the second half of the 20th century and physics was in the first half of the 20th century. That how our mental lives come to be from the interaction of 10 to the 10th neurons inside the brain seems a miraculous phenomenon, and we want to know everything about that we can possibly know about that. This is sort of drawing of the brain, viewed from the side and viewed from the top. I don't know if it's possible, this should be focusable a little bit. I don't know if it's possible. Do I have control of the focus at all? It doesn't matter so much. Anyway. Well, I think it's actually a problem with focus more like. Yeah, I mean, it's this bright English sunshine. Oh, there, that's better focus. Okay, that's a little bit better. Good. It's this bright English sunshine. The brain has a number of lobes that have identifiable functions, and we are busy studying the brain with all sorts of approaches. As many of you know, and as Graham alluded to the other night, there are now imaging approaches that allow us, at least with low spatial and temporal resolution, to image the brain of conscious awake humans. But there's also traditions of using electroencephalograms, or EEGs, which allows you to measure with much finer temporal resolution, follow the brain waves much faster than imaging techniques allow you to do, but with much cruder spatial resolution, so you can't tell exactly where in the brain they're coming from. And then, of course, neurophysiologists like myself use microelectrodes. Now, this is maybe a little bit much just after breakfast, but this is actually the brain of an awake and conscious human patient. And Wilder Penfield, a neurologist or a neurosurgeon at the Montreal Neurological Institute in the middle of the 20th century, actually showed that you could do this kind of surgery and expose the brain of conscious human subjects just using local anesthetics to deaden pain of the dura. And this kind of thing now is done here in Britain and at major medical centers around the world every day. Okay, so Penfield originally did it for treatment of epilepsy, incurable epileptics for whom the drugs of the time were not helping enough with the epilepsy, and he could go in and actually excise small pieces of the cerebral cortex, get rid of the damaged part of the cortex that was causing the epileptic seizures, and frequently make these people's lives considerably better. Today we do this kind of surgery mostly to implant microelectrodes into the brain for treating Parkinson's disease. Some of you have heard of deep brain stimulation, and increasingly this is being used to treat cases, or experimentally at this point, used to treat cases of severe depression and sometimes intractable pain as well. This is a sort of modern version of Penfield's thing, where Penfield used to put little ball electrodes down on the surface of the brain, but now neurosurgeons will lay a whole array of large electrodes on the surface of the brain, and each of these metal stubs is an electrode that's connected with amplifiers by these little wires that run through. And you can actually record the electrical activity of the brain in a very localized manner, or you can electrically stimulate it through these electrodes. So you can drive a pulse of electrical current into the brain, cause the cells to fire their little electrical impulses, the action potentials. And Penfield discovered that when he did this, that just by stimulating single places on the brain, he could elicit very organized mental experiences. So particularly in the temporal lobe, that we know plays a large role in memory, he could actually electrically stimulate the brain, ask patients what they felt, and patients would say things like, I have a sense of deja vu, I'm on the porch on my mother's house and I hear her calling me to dinner. Or I hear a band, it's a parade, and I hear a band playing some specific tune marching down the street. And these were very high levels of the cerebral cortex where you would elicit these organized mental experiences. But Penfield established then that organized mental experiences could result just from the electrical activity of the brain. You didn't actually have to have inputs from the sensory periphery. It could come just from the electrical activity of the brain. Now, these things are being studied on all fronts very aggressively. Neuroscience is many undergraduates are becoming progressively more fascinated with neuroscience, are coming to neuroscience in graduate school. And this will be a very rapidly moving field. And I want to talk today about two challenges of the neurosciences. And I want to talk just a little bit about the phenomenon of consciousness, which came up in the panel discussion last night, someone asked me about, and how can we or not potentially understand how consciousness is related to electrical activity of the brain. And what I want to address here is just the question of, is the whole more than the sum of the parts? Is it possible to reduce consciousness and understand it just by understanding connections in electrical activity in the brain? Or is there something larger than this that is going to elude our current approaches to science? And I'll tell you a little story from my lab, because we bumped up against this problem in my lab, actually. And the second thing I want to talk a little bit about is the issue of reductionism and the causal status of the mind. And of course, these two are not unrelated to each other, but they're a bit different. And this is the whole issue of determinism and to what extent we actually have choice control over our own actions. And that's what I mean by the causal status of the mind. And I'll tell you that the Christian aspects, specifically religious aspects of what I will talk about, are more implicit than explicit here. These are issues that are major for all of society, I think, in talking about human behavior and the biology underlying human behavior. And I think they're important for Christians and other religious persons to think about carefully. But my own perspective on this will be from a biological perspective, is a person who's both religious and tries to take biology and neurobiology seriously, and so it will be more implicit than explicit, though in the question and answer period, inevitably with a group like this, those considerations will become explicit. So let's talk a little bit about this consciousness here, and conscious awareness, and this rich internal life that we have. You know, I tell medical students at Stanford when I stand up to talk about vision, for example, vision cortex. I give the vision lectures because I do research in vision. And I tell them, you know, to think about your experience, your apprehension of the visual world. And all of you guys looking out right now and looking at me, listening to me, you feel like you have a direct apprehension of this room, of that door, of that sign, of the things, the words up there, and this immediate, direct apprehension of experience that we have. And yet that experience is created inside the brain. And the brain has no direct access to anything out there in the world. The only thing the brain knows about the visual world is the electrical impulses coming up from the eyes in the form of 3 million nerve fibers carrying these electrical impulses. That's it. Okay? That's the only visual information the brain gets from the world. You have 1.5 million ganglion cells in each eye projecting axons into the brain, and each of these 3 million lines is carrying a stream of action potentials, and out of those 3 million streams of action potentials, the brain creates this subjective reality that we're all experiencing right now. And I told medical students, you know, this is a near miracle. I mean, I will be telling you some things about the mechanisms, but how this conscious awareness, how this conscious experience gets put together or emerges from these 3 million lines of action potentials, I tell medical students, I can't answer that for you in this course. And it may be, you know, it leads you to wonder whether there will ultimately be fundamental limits in how we can understand consciousness based in neural activity. And this is a very famous essay written by a philosopher named Thomas Nagel called What Is It Like to Be a Bat? And I would recommend that you Google this thing and read it at some time if you're interested in these issues. And Nagel's thinking a lot about philosophy of mind, and he asks us all to contemplate this question: what would it be like to be a bat? So to be flying around in the air and to understand the world and navigate through the world not by the normal auditory mechanisms that we have or through vision that we have, but by sonar. So emitting little beeps and getting these things reflected back, doing calculations based on time shifts and localizing objects. And what would it be like to be a bat? What would it actually be like? What would that conscious experience be like? We know what our conscious experiences are, but what would it be like to be a bat? And Nagel says, and argues compellingly, in my opinion, that we have no clue what it would be like to be a bat. And that furthermore, it's difficult, if not impossible, for us to imagine ever learning that. And we can learn how the bat's brain works, and we can learn the mechanisms that the bat uses, but what it would be like to have those mechanisms inside of our own brains, we don't know. And Nagel says, he draws a larger point from this simple mental exercise. He says, if the subjective character of experience, so that's the way we really experience the world, is fully comprehensible only from one point of view, and that's the view of having those mechanisms inside ourselves, inside our own brains, and understanding, really feeling what it would be like to be a bat, then any shift to greater objectivity, that is less attachment to a specific viewpoint, does not take us nearer to the real nature of the phenomenon, it takes us further away from it. And of course, this shift to a greater objectivity is what modern natural science is all about. That's what we do. We provide third-person descriptions of these events. We provide descriptions that we can measure in laboratories, and that we can write our little method sections, and that any scientist anywhere in the world can make the same measurements if they want to. So modern science is all about third-person descriptions of events. But Nagel is saying here there may be something very unique about this internal experience of conscious awareness that is only appreciable, it's only fully comprehensible from one point of view, and that is the subjective point of view. That's the first-person point of view. And if that is true, then any shift to greater objectivity, no matter how much science we do on it, the shift to greater objectivity takes us further away from the real phenomenon, not closer to it. And that's a very important point to make. And this point is very controversial. Neuroscientists don't like to hear this. Neuroscientists see themselves as this great band of warriors who ultimately are going to conquer and understand all of this stuff, and nobody wants to hear that there may be some things that are quite difficult for us to do. But increasingly, there's a minority of thoughtful people who take this kind of point of view. Here's another quote from John Searle, a philosopher. This is not the John Searle here in Britain that many of you know. This is John Searle, philosopher at University of California, Berkeley. He wrote a book a few years back called The Rediscovery of the Mind. And Searle is very characteristically strongly spoken. And he says, much of the bankruptcy of most work in the philosophy of mind, and a great deal of the sterility of academic psychology over the past 50 years, have come from a persistent failure to recognize and come to terms with the fact that the ontology of the mental is an irreducibly first-person ontology. So irreducibly first-person. You get past the first person, try to go into the third-person description, and you inevitably lose some important stuff. Now, mostly, neuroscientists are happy to leave this kind of discussion to philosophers and let them sit in their rooms and have their seminars and talk. And meanwhile, we'll be about the business of solving the brain, okay? And we'll show them. But in my laboratory, I want to tell you a little scientific story from my lab, because I think we've actually run up against this problem in my lab and can come to a bit of a blank wall in a way. I mean, we did some really, really good science, I think, to get to there, but we came up to a bit of a blank wall and just had to go off in another direction. And I think this is the blank wall we hit. I think it's real. I don't think it's speculative. I think we've actually hit it to some extent in the laboratory. And I can just take you through a little bit of science and I think help you. I think everyone here, this is not difficult material conceptually. It's difficult technically to pull off and set up, but it's not difficult conceptually. So I think if we do a little review of high school biology here, is everyone up for that? A little review of some high school biology, we can get there. You can see exactly how we ran up against this. So just to review the biology, these are nerve cells, okay? And to some extent, they're like every other cell in the body. They have a nucleus that John and Graham have talked to us about, where a lot of the plumbing and machinery is done. They have this array of processes sort of on the outside of their field, and these processes are called dendrites, and this is where they get synaptic inputs or chemical inputs from all the other cells around. And then the action potentials, these little electrical signals, are fired right here near the cell body, and then they travel down the axon to little structures called synapses, where they release chemical transmitters onto the next cell down the line. So they take input through their dendrites and they send output through their axons to the next cells, and each one of these cells receives synaptic connections from about 1,000 other neurons in the brain, and in turn sends its own synaptic connections to 1,000 or so other neurons. So it's a very complex system. And if you just make a slice through a brain like this, you can make very thin slices of brain tissue and you can stain them so that you can actually see those cell bodies. This is what the cerebral cortex looks like. So this is the gray matter that lies on the outside of your brains, and every little purple dot in here is an individual cell, and you can see that the cortex really is a sheet of gray matter, that even if it goes way down in these folds, it comes back out on the surface again. It's like a crumpled up ball of paper, but it really is it's this sheet, and it's folded up to get more surface area, cram more surface area inside your brain. And if you take a little slice of this and blow it up further, you can see the individual cells. Each of these dots is a cell body, but you don't see the axons and the dendrites. You have to use a different kind of stain to see them. You can see these layers here that are a prominent feature of the cortex. And the layers have a lot to do with the inputs and outputs for the cortex. There is also vertical structure that you can't see in histology like this. You can only reveal physiologically by recording the electrical activities. And these vertical structures are called columns. And inside one column, like what I'm drawing right here with my laser pointer, that's about 100 microns in this dimension, about 100 microns in this dimension. There's about 1,000 nerve cells stacked up inside one of these 100 micron by 100 micron columns. And this is the microelectrode, which is the neurophysiologist's tool of trade. And this is a very thin piece of metal that actually has thin insulation down to within 10 microns of the tip. And the only uninsulated part is right down here. And if you get that uninsulated business end of the electrode up close enough to a nerve cell, you can actually record the action potentials from one nerve cell or a cluster of 2 or 3 nerve cells without getting too much noise from surrounding cells. So you can actually listen in to the traffic of a single nerve cell. Or, like I said, you can push little electrical currents through this microelectrode, and you can artificially cause activity here. So you can listen to the activity, or you can inject artificial activity, basically. And these microelectrodes are very thin. You can't even see this with your bare eye. You have to look under a microscope to see the business end of it. And these microelectrodes are put into, as I said, into people's brains all the time. There are tens of thousands of people now running around with these electrodes in their heads. And it doesn't hurt because there are no pain receptors in the brain. Okay, so we put these microelectrodes inside the heads of monkeys in my laboratory. But again, it doesn't hurt the monkeys because there are no pain receptors in the brain. That's actually a bad thing about the brain to some extent. It's one reason why brain tumors are so dangerous. The first sign you get frequently of a brain tumor is epileptic seizures or neurological symptoms like slurring of speech. If you have normal pain and inflammatory mechanisms there, you could detect them much sooner, almost certainly. I don't know why that is actually, but that is the way that it is. Okay, so we do this work that I wanted to tell you about that we get up against this wall in the lab. Has to do with our work with monkeys, and we work with rhesus monkeys. They're macaques, and you can train these animals using operant conditioning techniques, just like you do in zoos. You train animals in zoos by giving them a rule or something they want to work for, and in turn, they will sit and essentially play video games for you. So they'll sit there and look at a visual display in a given day and tell you what they're seeing. We train them, and I'll let you do a task in just a moment that our monkeys do, and we'll see if you're as good as the monkeys are, okay? And this is the monkey's brain here. This is the front of it, this is the back of it. We have taken the cerebellum and brainstem out of here because this is the cerebral cortex that we are most interested in. And at the back of the animal's brain, at the back of your brains as well, is the parts that do vision. So these are the parts that I've worked with historically, mostly in my lab. This is the first visual area. But there are a lot of other secondary visual areas, or so-called higher visual areas. And one of them that I've worked with a lot is right here in the middle, this middle temporal visual area, or MT. And the reason I've worked a lot with that is that we believe it's a pathway that's dedicated primarily to processing motion information. Now, what do I mean by that it processes motion information? What I mean is that the action potential discharges, these electrical discharges of the cells, give the rest of the brain information about the motion of stimuli in the environment. So if you take a stimulus like a bar of light, and you just project this onto a screen or you present it on a television screen, and you move it across the visual field in one direction. You can't see these things here because of the bright sunshine. But these are little vertical red tick marks that indicate the discharge of an action potential that we can measure through those microelectrodes. And if you move this stimulus across the visual field in one direction, you get a lot of these action potentials being discharged. But if you move it back in the opposite direction, this is the same stimulus across the same part of the retina, but in the opposite direction, you only get 1 or 2 action potentials. And so this kind of cell, by virtue of the way it's hooked up to the retina and is processing information, seems to capture information about the direction of motion of stimuli and broadcast that information to the rest of the brain through its action potentials that go out. Now in this area, well, I'm going to show you actually what directional selectivity sounds like. So this is a movie that was actually taken in my lab, and what you're going to see on the screen is what the monkey sees. So this camera is behind the monkey's head, and you're going to see what the monkey sees on the screen. And what you're going to see is a field of random dots, and you're going to see these dots moving first in this direction down and to the left, and then they'll move in this direction, and then they'll move in this direction. And we take the signals from the microelectrode and we amplify them with standard electronics, and we play them out on a loudspeaker, and what you're going to hear on the soundtrack is pop, 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 and every pop is an action potential occurring from one of these cells. And your job is to listen to the action potentials, and you'll see that the action potentials, the rate at which they're discharged, vary as a function of the direction. And this is classic direction selectivity. So you'll also hear, before a stimulus comes on, before the random dots come on, you'll hear the pop, 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 because these cells fire spontaneously to some extent, and it's only the rate that gets picked up when we show the stimulus. Now, I don't have speakers here, but I think this is a small enough room that you can hear this, but you'll have to listen pretty carefully. So there's the spontaneous activity, pop, 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 pop. So that motion was going down and to the left. Now it's down and to the right, and it's a smaller response. Now it's directly to the right, and it's a smaller response. And when the motion goes that way, the cell's completely silent. Now it's going up and to the left. Now we'll go directly to the left. There's directly to the left, and the cell's picking up again. And that's what direction selectivity is like. So that's deep in the brain. It's deep in the middle of the brain. And you can actually hear, you can just listen to it, and you can tell what stimulus is on the screen just by listening to the nerve cells. You don't even have to look at the screen. And so these cells, whoops, that was not what I wanted to do. These cells are directionally selective. And then I have to tell you this one other thing before you can really appreciate the experiment. The one other thing is these cells are organized in clusters. So a cell that likes upward motion, a cell that likes rightward motion and leftward motion, they're not just randomly put together willy-nilly. They're actually organized in separate spatial clusters. So we can actually put an electrode into one of these cells, into one of these clusters, and we can electrically stimulate, say, a cluster of cells that likes downward motion, or we can stimulate a cluster of cells that likes upward motion, or rightward or leftward motion. And we can sort of ask the animal to tell us what he's seeing when we electrically stimulate these cells. So the way we ask the animal what he's seeing is we ask him to do a direction discrimination task. And this is the place where we're supposed to do this and see if you can do this as well as the animals. You're going to see these random dot patterns, and I really wish we could make it darker in here.
We can just close some of these screens.
Any bit that we can make it darker, at least temporarily, we help. That's where the biggest problem is coming from over there, and that helps. That actually helps. That's going to help a lot. Okay, so you're going to now see these patterns of random dots, and your job is to tell me whether you see upward motion or downward motion. And don't be shy, just yell it out. And so, so tell me what direction you see when you see this motion stimulus. What do you see? Very good. Okay. So what direction do you see? Good. So right now, 50% of these dots are moving coherently, and 50% of them are just flickering. Okay, so that's a strong motion signal, and all of you can do that. Now we'll drop this to 25% of the dots. Which direction did you see?
Left.
Down. Somebody said left. Direction?
Right.
No, it wasn't right. It was down. It was down. Now we'll cut it to 12%. So some of you may still be able to see. Up. Yes, this is up. Direction? Yeah, that was down. Correct. Direction? Yeah, that's up. Now we'll cut it to 6%. You can't win in the end. I'll keep cutting it down. Okay, so don't be. I think that was probably down. I'm not completely certain. Yeah, I think that was up. I heard it down, but I'm not so sure. Anyway, you get the idea. So the way that the monkey does this is just to sit and look at these things, and he'll hit one lever or another lever. And if he gets it right, we give him juice, Kool-Aid or Tang, or we try to find a favorite juice for each animal. And we'll give them the shot of juice in their mouth for a correct answer. For you guys, we might give you, a shot of Tetley's Ale or something like that. So this is the direction task that we asked them to do. And then, like I told you, if you ask my colleagues, and actually I think one of my colleagues is here today, who's working in a physiology laboratory here at Cambridge. If you ask my colleagues around the world, Newsome's done a lot of good work over the years, all those papers that Dennis mentioned, but the one experiment that was most unexpected and in some ways got the most attention, it would be this experiment. Well, we actually had monkeys doing this task, and they would stare at the dots and they would try to tell us, is the motion upward or is the motion downward? We would actually electrically stimulate a group of these downward cells. And the question is the electrical activity of these cells really supplying the information that the monkey uses when he makes a judgment like this? So the whole monkey is sitting there looking at these dots and trying to judge whether it's up or down, and we're electrically stimulating a little cluster of cells deep in the brain that's only about 100, 200 microns in diameter, something like that. And what we showed is that if we stimulate these down neurons, we can systematically bias the animal toward judging and reporting downward motion. And if we just move that electrode 100, 200 microns and stimulate up cells instead, we can bias the animal toward making upward judgments. Now you guys remember that movie, those very first movies that I showed you where there were 50% dots and everybody was getting this right. You can show the monkey that exactly same stimulus where 50% of the dots are moving upward, but if you stimulate these down columns, the monkey will report seeing downward motion. And we've documented that. I could show you quantitative data more, it would nauseate you how much data we have over this, and I'm going to spare you that. I'm just telling you the actual result. But now this raised a question. This is where we hit the bump, okay? This is where we hit the wall. And the question that it raised is, what is the monkey's actual subjective experience when we stimulate MT? If the monkey could talk to us the way we can talk to each other, what would he say? Would the monkey say to us, hey guys, I saw downward motion on that trial, and I reported downward motion. Why didn't you give me a reward? Or would the monkey say, I saw upward motion on that trial, but I reported downward. I don't know why I did that, right? So the behavior that we can measure is consistent with either of those interpretations. And what I'm really wondering is, what is the animal's actual subjective experience? Does the animal actually believe that he sees motion in a particular direction? Is he seeing something that I don't know about? Are we changing the decision somehow without actually changing the basic percept? And I don't know the answer to that question. And furthermore, I don't see how I can get the answer to that question in my laboratory. We thought about this, and there's potentially some things we could do. Different clever psychologists have suggested different things, but they're all basically ways of trying to make an end run around the basic problem. And the basic problem is that I have no way to monitor that animal's first-person experience. Okay, so that creates a problem, and it creates the problem that I want to know, what does the monkey see when MT is stimulated? And the more I've thought about this, the more I think the gap is there, and the only way ultimately for me to know this is to stimulate my own MT, create the first-person experience inside my head during the electrical stimulation. Now, given the fact that you and I have similar brains and we have a shared vocabulary and we've developed a system of words to describe shared experiences, maybe I could communicate what I saw to you in the form of a written journal paper or a talk like this. I don't know. But if it were something truly unique and novel, I might not be able to communicate it to you. So this creates a paradox for me as a neuroscientist. And the paradox is that I can envision a day 100 years into the future, 200 years into the future, however long it takes, when we have a really mature neuroscience. I can envision something where this entire loop is understood in electrical, physiological terms, where we know exactly how that pattern of dots stimulates the cells on the retina, We know all of the action potentials coming out of this thing into the brain. We know them on a millisecond by millisecond, nerve fiber by nerve fiber basis. We know exactly how the cells in the visual cortex are reacting. We know how that animal's forming its decision in third-person objective terms, in cause and effect, and just in terms of the way nerve cells affect other nerve cells. We know how he plans his operant response and how he ultimately selects it. We know all about reward and motivation systems inside his brain. I can envision a time where we know all this in terms of the third-person description, the causal loops that process information in the brain, and still may not be able to answer the question, what does the monkey see when MT is stimulated? Right? Now, that creates a problem. If we know the whole system from input to output in electrophysiological terms, but we can't answer this question, is there more to be discovered or not? And some neuroscientists would say, no, there's nothing more to be discovered. If you know the whole thing from input to output, and you can predict the output based on the input, then this is irrelevant, and we just move on to the next problem. But I think this is why I'm in neuroscience. This is what I really want to know. I really want to know how mental experience arises from these systems of neurons, and if we get to a point where we think we know all the answers, but we can't answer this question, I would say we don't know all the answers. And so in this sense, I would say that conscious experience and the nature of first-person awareness is a profound mystery. And one of the mysteries is, why should conscious experience result from the electrical activity of nervous systems at all? Why is it that synapses and action potentials and interactions in a complex system, why should we have this internal feeling that it's like something for me to see? I don't know the answer to that question. And I think there may be fundamental limits on what we can understand about conscious first-person experience using the third-person methods of contemporary science. We may have to invent a new science. Maybe one day there will be a science where we can do something like objective science on first-person experience, but this is a very difficult problem. So that question I think you asked in the discussion last night. This is a much more detailed answer than I was able to give last night about my own views on this. And we can talk about this some during the question and answer period. Okay, so let's go on to this second problem here, which is very important for us about reductionism and whether or not all of our actions are determined by the physiological activity of cells in the brain. And how are we to think about human autonomy and human freedom and human moral responsibility? So, of course, neuroscience, our program is to try to understand mechanisms. It's try to understand mechanisms of the brain the same way a local mechanic understands mechanisms of an automobile. But in the end, all of our legal systems, all of our moral traditions, our religious traditions depend upon being able to think about and take seriously some freedom of choice and some responsibility for choice of human beings. And this is going to be inevitably a collision area between standard reductionist neuroscience and our religious, moral, and legal traditions. So let's think a little bit about this. And I don't have good answers to this. I mean, this is the problem, the old problem of free will, and it's been with us for centuries, and I don't have good answers to it. I don't have compelling answers to it. But let me just tell you some of the things that I thought about, and maybe I can at least kickstart a discussion. Dennis, I don't have a watch on. Can you tell me how much time?
You have 16 minutes left.
16 minutes. Okay, I think we can do that.
More or less.
More or less. Yes. I'll handle the problem of 30 centuries of human reflection in 16 minutes. Right? Okay. So this was a comment on reductionism. I'm not a fatalist, but even if I were, what can I do about it? Yeah, that gets better the more you think about it. So John Searle, this philosopher of mind who I alluded to earlier, basically in his little book, he published this little book called Mind in 2004. It's a nice little summary of contemporary thinking about philosophy of mind. It's easily readable in about 3 hours. And he wrote this book, and when he came to the problem of free will, he said this is the one that's most vexing. And I only see two options for looking at things. Hypothesis one is to go ahead and accept determinism. in the mechanical brain, and that our notions of freedom, our notions of choice are basically illusory notions, and that what's really going on deep inside our brains is exactly the same thing that's going on deep inside of a car, and there's no freedom to it whatsoever. And he said the only way he sees to escape determinism is by talking about quantum mechanics and the quantum brain. And there are some very smart people who believe that quantum mechanics can allow us to somehow create mechanisms inside the brain that are not characterized by determinism. Now, I personally am not terribly attracted to either one of these things. So quantum mechanics is fundamentally describes, it's our most basic theory of physical reality now, and it describes things probabilistically. It says that at a microscopic level, where microscopic is very, very small indeed, that events can only be predicted probabilistically, and that within the probabilistic framework, events happen randomly, and that's how many people seek to escape strict determinism. I don't find this very attractive. I want to take responsibility and choice seriously. And I don't find quantum mechanics a very attractive alternative because fundamentally quantum mechanical events are random events within certain probabilities. And I don't find randomness any more meaningful than complete determinism. Okay? So when I'm crossing that street out there later this afternoon, I don't want a probabilistic, I don't want to say, my brain is reliable at identifying the oncoming car with 99.9999% accuracy, but occasionally you won't see the car because then I'm dead. I mean, that's not a good thing, and I don't like this very much. So I think that the perspective that is most useful from my point of view, though it's quite tricky, is this notion of emergence in complex systems. And a lot is being written about emergence these days, and I'll make a recommendation to you of a paper at the end of this talk that I think talks about it as clearly as it's about possible to talk about it right now. And what is emergence? Okay, emergence has two, in my personal definition, has two prongs to it. One is the complex systems, like a brain, for example, but like many other, just like a living cell is another example of a complex system, they can behave in ways that are not predictable from knowledge of the components alone. So we can have all the knowledge that we want to have about individual molecules, but before a cell actually comes into existence and we observe its behavior, we could not predict how it would behave. And I feel the same is true about brains, ultimately. The knowledge of the individual components of individual neurons alone is not enough to predict how the system is going to act when it's together as a system. And this is very critical to me. These systems, complex systems, are governed by rules that are independent of those that govern the components, although they're constrained by them. So an emergent system cannot violate the laws of physics. It's constrained by the laws that govern the components. But these systems exhibit behavior and are governed by their own internal logic that are independent of those fundamental laws of physics. They're constrained by them, but they're not determined by them. So if this is true, then the standard reductionist paradigm, and I would say that the standard reductionist paradigm says that when we can break a complex system into its components and understand the mechanisms underlying it, then we can, in some senses, abandon the high-level description because we've reduced it to a more fundamental level. And I would say that this standard reductionist paradigm is impoverished. And it's not wrong, it's just impoverished, okay? And it's impoverished in the sense that it does not appreciate these rules that are independent of the lower components. Now, I can see and I suggest maybe this kind of poverty lies at the heart of some of our difficulties thinking about free will and other emergent phenomena. So I can see the thought bubbles over many of your heads right now, and you're saying, what on earth is he talking about, right? And I'll try to give you some examples. I'll work through one example in a little bit of detail, but then I'll give you some more casual examples once we get the principles down. So the example I want to work through in some detail is the example of neural networks. So many of you, I'm sure, have heard of neural networks, and these are creatures of the artificial intelligence movement. And neural networks actually, their predecessors came into being in the 1960s, but the modern era of neural network research was kicked off in the 1980s with the publication of a big series of books edited by Rummel, Hart, and McClellan. And this is basically how these things work. These things run on digital computers. You can run them on your PC at home or your laptops or whatever if you want to. You can buy software packages now that allow you to create these things. And neural networks are made of little processing units. So each of these circles is a processing unit. And these processing units are neuron-like elements, and thus the name neural networks. And the simplest but very sophisticated neural networks have 3 layers to them. They have a layer of input units, so these are your input units, and then there's so-called hidden layers or internal representation units, and then you have outputs, okay? And each one of these units is connected to every one of the other units in the next layer. So if you look at this guy here, he's got arrows going to each one of these. So he has inputs, he has connections. The activity in this unit here will influence all of these units here, and in turn, activity in this unit will influence all of these units here. Now, the amazing thing about neural networks is that you can code a certain kind of inputs. You can give them particular inputs, and you can set a neural network a very difficult problem to solve. And these things were most spectacular in the 1980s in the problems of voice recognition and character recognition. So you would set something like presenting visual images of letters, a sequence of letters to the input pattern, and asking some voice synthesizer to pronounce those letters at the output. Now that's an incredibly difficult problem. Years and years of artificial intelligence and coding by traditional algorithmic methods has failed at this problem nearly completely. And yet in the 1980s it was shown that these simple neural networks could solve this problem and put out reasonable speech in response to letter input. Now this was actually amazing, and you say to yourself, how did this happen? The way this happens is that this network has the ability to learn, okay? And the way that works is you give it a little input, and you look at the output and you say, okay, was it the right answer or the wrong answer? Or how right or how wrong was it? And what happens is, depending upon whether it was on the right track or not on the right track, you go back and you modify these weights. So this little unit has these arrows going to every other unit, and how much this unit influences that guy or that guy or that guy or that guy is determined by the weights on these connections. And these weights can be near 0, meaning that this guy has no influence on this guy, or they can be very large, meaning that this guy has a lot of influence on these guys. So the learning comes into play by modifying these weights. And these things can go and learn on their own. So you program this thing and you compare the desired output to the input that comes in, and you write a little computer program to do that, and then this thing goes and iterates this over 10,000 iterations, modifying these little weights between this layer and this layer and this layer and this layer, and sure enough, it learns to solve the problem. And these things are amazing because they were doing things that human programmers could not do and still can't do in many cases. And this raises an interesting situation because you ask one of these programmers, they showed you these things. I remember when I was at a scientific meeting and saw this kind of stuff for the first time, and they look at this and they grin at you and they say, this solves this problem. And it's an amazing problem, and you look at them and you say, that's amazing. How did it do it? Okay? And you say, how did it do it? And they tell you that this is the answer. This is how it did it. And what this is a representation of those weights. Remember those weights that get learned? And a big white square means that's a large positive weight between one unit and the next unit, and a big black square means it's a large negative weight, and little squares means that there's no weight at all, so that unit doesn't influence the other unit. And you ask them, what's the answer? How did it do it? And they say, that's the answer. And I remember I didn't know whether to laugh or cry when I first heard that. My reaction was, that's not an answer. But in a physics sense of the word, it is an answer because cause and effect is known completely inside this network, okay? So the network is totally understood in one sense of the word. We know exactly what those connections are, and we can take that thing from one computer into another computer, and we can transmit that solution anywhere in the world. So it really is a solution. And in a physics sense of the word, in a cause and effect sense of the word, how one unit affects the other unit, there's nothing mysterious here at all. We totally understand this thing. But it's deeply mysterious in another sense, in the sense that we frequently, as humans, we don't know what the algorithmic solution is that that network has learned. And we sense that it's not enough just to know this. If we really want to understand the network, it's not enough just to know this, but somehow we want to know algorithmically what strategy that network has learned in order to reach this solution. We want to be able to ultimately write some kind of equation for it, write some kind of reduced description of it. And that is a gap in our understanding that we get this, I at least get this creepy feeling that network has discovered something and it understands something that I don't yet understand. So this to me brings into dramatic relief this notion of a gap between physics level understanding and the kind of understanding that we really want at the end of the day. And I think when we hit that kind of gap, that's the signature of an important emergent phenomenon. And one thing that I want to emphasize here, I'm trying to be faithful to my biology and I'm trying to be faithful to my science in that there's no magic here and there's no dualism. I'm not postulating any kind of living spirit inside that network in a sense. I'm not postulating magic, I'm not postulating anything that can't be understood because in terms of physics we know that network and I'm not postulating anything mysterious at the physics level. I'm not saying that physics ceases to exist or physics ceases to govern the network. But there is a gap here, and that gap is more common than we realize. So I would say, for example, cells, you see the same kind of gap. When the first cells came into existence in evolution, new phenomena came into existence that could not have been predicted from knowledge of molecules alone. What are those phenomena? Well, motility, for example. Cellular motility came into existence. The cell moves from one place to the other place. That concept, the concept of motility of a cell had no meaning. It just was not meaningful until cells came into existence. Metabolism, signal transduction, all sorts of cellular phenomena and logic of how these things are related to each other came into existence with the first cells that are an emergent phenomenon. More emergent phenomena came into existence with complex organisms. For example, predator-prey relationships. How can you ever understand predator-prey relationships in terms of molecules and DNA and proteins? This is an emergent phenomenon, predator-prey relationships, that only emerges with the emergence of complex organisms in evolution. There are other concepts like symbiosis, two organisms living fruitfully with each other, or parasitism, or what John talked about last night in his lecture, cell fate. In unicellular organisms, the cell just divides. All cells have the same fate. But in complex organisms, one cell becomes a liver cell, one cell becomes a lung, one cell becomes a brain cell. That concept, it just didn't exist before complex organisms came into existence. And I think computers are the same way. So in one sense, if you want to know how this computer operates, you can ask questions at the physics level. You could talk about the transistors in here and the electrons and the voltages and things like that, and you could understand input to output in terms of the physics of the transistors inside that computer. But I would say that if you don't understand what a for loop is, and that's a software concept, right? If you don't understand what an if statement is, if you don't understand what subroutines are, and this is an emergent phenomenon, crashes that we're all very familiar with. These higher levels of understanding are certainly things that we have to get to that are, again, not determined by the physics. An if statement is not determined by the physics, but it's constrained by the physics. If statements and subroutines can't do anything the physics don't allow them to do. So I would say that at the very least, this kind of gap, this emergent gap, is epistemological. In order to understand the neural network, in order to understand the cell, in order to understand the computer, we have got to get to these higher levels of organization that have to be understood on their own. But, here's the deep question. Is it ontological as well, right? So what is the status of these epistemological gaps? And is it possible that the higher level system configurations, like the cell per se, or the organism per se, or the neural network per se, actually exert causal influence over the lower levels? And this is the concept in the emergence literature of downward causality. But none of this is new with me, by the way. I'm just packaging concepts that are out there in my own terms is all that I'm doing here. And if it's helpful to you, that's great. If it's not, that's okay too. There will be another talk, a better talk after coffee. So does the higher level system exert causal influence over the low levels? Now, I think you can see now how these concepts relate to the idea of the causal status of the mind. Okay? Even acknowledging that nothing about our behavior that gets produced in my everyday life can be inconsistent with basic cell biology and can be inconsistent with physics, there is still, I think, a way to think about higher levels of the system, like my conscious self, exerting downward causality over the rest of the system. For example, when I raise my arm, some higher level of my brain made a decision to raise my arm. But part of raising my arm was action potentials going down those axons and releasing transmitter at the muscles, and little tiny ion channels, little single proteins, opening up and allowing ions to flow into the muscle cells to cause them to contract. So a decision that I made at a very high level of my brain has control over single ion channels. It affects downward causality on single ion channels. Okay? So I think that that for me, when we start talking about how do we understand human freedom and human self-governance, that this kind of concept of downward causality is ultimately going to be important. Now, is that what we mean by free will? It may be. I don't think free will is complete bottom-up determinism, but I don't think it's randomness. Okay? What I think is meaningful to free will is the concept of self-determination. That some higher level of my nervous system can, to some extent, determine its own activity and exert downward influence over the rest of my body. Now, this is another critical question for me, and I don't know the answer to this. Is this science or is it philosophy? So is downward causation, is it a philosophical concept that allows us to understand better, or is there actually a science here, a science of downward causation? And I don't know the answer, but there are some signs out there that it might actually be turning into a science. This is a book written by a guy named Scott Kelso, Dynamic Patterns, the Self-Organization of Brain and Behavior. And this is based on nonlinear dynamic systems, which is an area of physics that the physicists in the room will know something about. And people who study dynamic systems are familiar with this idea of downward causality. And Kelso, they talk about order parameters. There are these order parameters, that high-level order parameters that govern the behavior of dynamic systems. And Kelso says an order parameter is created by the coordination between the parts. So there is bottom-up influence on the order parameter, but in turn, it influences the behavior of the parts. And this is what we mean by circular causality. Now, I hope that's not the same as circular thinking. But I like this idea of circular causality, that there is bottom-up causality, that to understand physical systems, real physical systems, then we have to understand the bottom-up causality. And of course, that's what contemporary science mostly concentrates on. But my hunch is that getting into nonlinear complex systems, that this downward causality and understanding these high-level parameters that exert downward causality are going to be important and may actually become a part of science itself. So I'll just close by saying that I haven't solved the problem of free will. I've just tried to give you some perspectives from my particular point of view. But as a neuroscientist, I do believe that understanding the nature of human freedom is the most important problem facing the neurobehavioral sciences, because it's critical for our notions of human dignity and social responsibility and our religious notions as well. But oddly, if that were the only justification, that would be strong enough. But for my reductionist scientist friends, I say that understanding human freedom is also important for science itself. Well, what do I mean by that? And what I mean by that is that natural science itself assumes that scientists are free to some extent to evaluate evidence rationally and make decisions based upon evidence. And J.B.S. Haldane, the great British biologist of the middle 20th century, kind of put this way. This is one of my two favorite Haldane quotes. It says, "If my mental processes are determined wholly by the motions of the atoms in my brain, I have no reason to suppose that my beliefs are true, and hence I have no reason for supposing my brain to be composed of atoms." So there's a circularity here. Trying to be a complete bottom-up determinist to me is like crawling out on a limb of a tree and sawing it off under, behind you. There's intellectual free fall. And I don't see a way around this. I don't, if this proposition is true, that mental processes are determined wholly by the motions of atoms in my brain, then why is it true? What are the rational bases on which you would evaluate some proposition and come to the conclusion that it's true, and then you don't have a reason for supposing your brain to be composed of atoms? This, of course, looks a lot like postmodernism, right? To me, this is an irony that if I were an ardent determinist, I would also be an intellectual bedfellow of the the ardent postmodernist. But since I'm a natural scientist, I'm not a postmodernist. I believe that the statement that the pie is in the refrigerator is not just a power structure composed by one social group for its advantage over another. But, it's an empirical statement that really can be tested. Is the pie in the refrigerator or not? I mean, some things are power, but not all. So this is the paper that I wanted to tell you. If anyone's really interested in this George Ellis has written a very nice paper. It tries to grapple with this whole idea of emergence and top-down causality with as much common sense and as much scientific integrity as in a short format that I've seen, and it's called Physics in the Real World, and it's available at this website here. So thank you for your time, and I'll be happy to try to answer any questions you might have. Yes, yes.
Keep the neurons firing there.
Yes, absolutely.
Yeah. Thanks, Bill, very much indeed. I'm sure that's raised lots of interesting discussion. Just deconstructing the pie in the fridge.
Yes.
I think we get to so who wants to kick off on the brain? Yes.
The term ontology related to gap. I wondered exactly how you understood that.
This is one, who was it last night who said, mind open and mouth shut or something? That was a Mackay quote read. I can't give you a really, the question to me is, so ontology is about reality, as I understand it. So in a neural network or in a living cell or in a brain, I am 100% convinced of the epistemological gap. That to understand what that neural network is really doing, it's not enough to have the complete understanding at a physics level, the weights, because that comprises a complete understanding at the physics level. I'm convinced that to understand what that monkey's MT is doing, it's not enough just to know the inputs and outputs of every single nerve cell. I really want to know more about what that animal's subjective experience is. So I'm 100% convinced about the epistemological gap. The real question for me is the ontological gap. What is the ontological status of the higher levels? Are they real entities that should be given ontological status? And if they have downward causality, then I think the answer is clearly true. They deserve ontological status. If they act in the world, and if there are consequences to their actions, then they deserve ontological status. And I think, that I would love to be, I want to be an ontological emergentist. I'm just not sure if I can get there yet. Because the big question about the downward causality to me is if it's causality, if it really exerts causality, then it should be physics. We should be able to write equations for it at some point. We should be able to do science with it. And if it's not, then maybe it's just words. Maybe it's just philosophy, and maybe the ontological status is in doubt. So that's as far as I've gotten so far, and I don't know the answer. Do you have thoughts about that?
No, but I was just interested by what you were driving at.
Yeah.
Can I just throw in perhaps, I don't throw any question or comment. I mean, do you think there's any possibility of getting a handle on consciousness, which would involve organizing modules of the brain that are assessing, your MT neurons that are firing, and a kind of, assessment networks? And if you had a handle on assessment networks, which were kind of, I'm not looking for a homunculus inside the brain, which is finally looking at the picture, which is the way that conversation can go. But,
right, exactly.
But if there are higher order networks which are kind of assessing what's going on at this sort of lower firing level,
yes.
I mean, and you can understand more about those kind of higher assessment networks.
Yes.
Would that, do you think, give any leverage that you could then wonder what the monkey is seeing or
Okay,
something?
so first of all, those higher level assessment networks definitely exist. And we've done research on some of them in my lab. So just MT, for example, this sensory area that has these directionally selective cells. When you look at those ambiguous dot patterns and you're trying to decide is it up or down, or when the monkey looks at that, those clusters of cells are in essence voting. The cluster of up cells is firing at some firing rate that's proportional to the amount of its stuff it sees out there, and the down cells are firing proportionally to the amount of their stuff. And the likely decision variable is probably in the brain. A higher assessment network just does a simple subtraction. It says which of those is firing the most, and if the up guys win, the monkey says up. If the down guys win, the monkey says down. That is an assessment network. It's a very simple assessment network, but it's an assessment network. And we call that a decision process, or we call it a decision variable being computed inside the brain. But there are much more sophisticated assessment networks, about whether I'm achieving rewards, whether I'm achieving goals. I mean, I'm assessing you guys all the time I'm talking. How many people are nodding off? Am I losing my audience? Do I need to be more lively? There are these assessment networks all the time. And yes, it will give us some leverage on, not only give us some leverage, it's absolutely essential to understanding higher brain function. However, the same problem exists. I can describe those assessment networks till the cows come home in third-person objective terms and perhaps still not understand the conscious experience that comes along with the activity of the neurons involved in those assessment networks. I mean, it is a deep problem. So philosophers of mind, people who do this kind of stuff for a living, and I'm not one of them, and I am only an amateur here, they talk about the hard problem of consciousness and the easy problem. And the easy problem, I think, is what you're talking about getting leverage on. It's about understanding mechanistically these processes that mediate self-awareness or self-reflexiveness, allow us to reflect about our own action. And I have no doubt that neuroscience will get to that point at some point. But then they talk about the hard problem, which is still, why should there be any subjective experience at all accompanying the electrical activity of neurons? What is that about? I don't know.
Yeah, kind of bring it into a pastoral framework.
Yes. Yeah, it's a long stretch between this and pastoral framework, I know.
15 years ago, I had a near-death experience, okay? And one way you could say, okay, I was taking whatever and had this experience. Another way you could say it's a neurotransmitter working my brain. In a sense, that doesn't matter if the experience that was interpreted after that event that was significant for me at that time, and others I know with a similar experience. Is that legitimate to interpret it? Does it in a sense has to matter than that you're talking to us about?
So say it again now. So you had an experience 15 years ago, but you say the important thing is not so much the experience as the narrative that's developed since the experience, the way you've interpreted it. Am I hearing you right?
It's more the sense to me of peace, of God's presence, or whatever, that was very real to me at the time.
Yes.
It could be interpreted, I guess, physiologically in one sense, or at the time, spiritually. In a sense, that my reflection since is that doesn't matter. What matters is the way it took my relationship with God.
Yes. Okay. So I agree completely with that, and I tried to say that a little bit last night. You guys remember in the panel discussion where I gave the example of color vision, that we know a lot about color vision, about the mechanisms that in the retina that give rise to our color systems, about the physiological mechanisms, and we can describe that in third-person terms. But that third-person description, to me, will never substitute for the actual experience of seeing color. So there's this story in philosophical circles, this little mental exercise about Mary the colorblind neuroscientist. So someone who cannot see color, but has studied color professionally, someone who's colorblind but has studied color and knows all about, in theory, the three cone mechanisms and how they lead to the perception of color of normal-sighted people, and know how those signals are processed in the brain. And the question is, this person has this exhaustive scientific knowledge of color vision. Would she learn anything should she suddenly become non-colorblind and see color for the first time? And I think she would. I think, to me, it's dead obvious that she would. She'd learn what it feels like to see color. She'd learn the first-person experience. But it's amazing how many of my colleagues disagree with that. So it must not be as obvious as I think. But what I said last night is that the third-person description will never substitute for the actual experience itself. So that experience of peace with God has its own vitality, it has its own reality. I think it has, you know, from the emergence point of view, it has its own causal reality in it, and it is the primary experience. If I had the choice between experiencing love with my family versus having exhaustive neurophysiological knowledge about what that love means inside my brain, as you know, as famous as that would make me, I would choose this one, right? So I completely agree that the fact that there are mechanisms, the fact that there are mechanisms, and I believe there are mechanisms, it does not substitute for the primacy of the experience. And in pastoral care or in our living of our own personal lives, the primacy of the experience is the important thing, whether it's the love of family, whether it's peace with God. I mean, you could view this. I think somebody asked me in London on Saturday, you know, why do I care about all this? You know I have free will. Why do I care about all this? And I said, maybe you don't. You know, there's some of us who are interminably curious about how the brain works, and we want to know about these things. But if you're not curious, then it doesn't matter.
Yes. Pardon the complete layman's view on it, but
well, I would ask you to pardon the confusion.
The kind of question that you posed about the monkey, in
Yes.
other words, was it really seeing it, or,
Yes.
or
Yes.
was it really seeing something different, but for some reason said it was?
Yes.
Is there any relation between that and the kind of confusion that we see pastorally with people with strokes, who might be able to read a word and tell you what letters the word were, but say a completely different word, and they don't know why they're saying it? I mean, as I say, it's very much a layman's question, but I just wondered if you can get any handle on on it from observing
I think
human.
I think that potentially you can, and you know the effects of strokes, which are sometimes highly specific, like you're describing, and deeply non-intuitive to us as observers. That demonstrates how sophisticated the mechanisms are, and it demonstrates that even the highest levels of our behavior about reading or interpreting or extracting sense are based on neural mechanisms, and they're in the brain, and they can be damaged in very non-intuitive ways that separate these functions out for us. So I think yes, I think that sometimes you can get insights into, you know, we have a whole experience of reading and interpreting and understanding, but that whole experience that we have is actually the result of several complex processes going on in parallel inside the brain. And you damage one of those things, and you can isolate it, you can dissect it, you can take it apart, like you're saying.
But you can't tell, you can't say on a neurological level what's happening yet in that sort of situation. Sometimes you can, sometimes you can't. The problem is on the neurological level, you can say those kinds of strokes can tell you that this part of the brain is deeply involved in this psychological or this formal computational process. But the problem is that you're limited to that. You cannot then, because in humans you can't do detailed neurophysiological experiments, you can't then go in and start taking it apart even further and say, okay, the neurons in this little part of the brain, how are they actually connected to other parts of the brain? What are their input signals? You can't really start taking it apart in deep physiological You're not allowed to.
You're not allowed to.
No, very
not allowed to, because it's risky.
Okay,
it's
so
just-
monkeys then.
Yeah, yeah.
So if you could do that with a human.
Potentially you could
Yeah,
start taking
yeah,
it apart.
yeah.
Potentially you could start taking it apart. Yeah.
Yes. I was interested in the sort of diagram where you began with sight and moved through decision and moved
Right.
through all the way around to motion and it being circular. But I can understand with sight, there's an external stimulus that begins the process.
Yes.
But if the process begins somewhere else, say with planning,
Yes.
what kind of stimulus causes that? And particularly if it's planning. Well, I studied architecture before I went into theology, and I would come up with these brilliant housing designs in my head that were not related to anything I'd seen before, and I could see it beautifully in my head. I couldn't draw it because I didn't have the artistic gifts, but where does that process begin?
It's a great question. It's a great question. So, right now I'm planning to walk down those stairs in a while, and that actually comes from some hunger that I have, because I didn't make it to breakfast this morning. And I want to get down to those cookies and tea. You know, they can come from very, very different places. They can come from memories that we have. And what the question that you're asking, to me, raises a question. It's a systems kind of question. So I tell the medical students at Stanford when I give them the motor systems lecture, you know, you can ask the same question about when I move my arm, where is the center that's responsible for moving the arm? And you know, you raise your hand and ask a question. What started that movement? Was it the motor cortex? Was it a curiosity center in the brain? Was it smart-ass center in the brain? You know, and that kind of question, it's a mistake. It's fundamentally a mistake for us to look for individual centers in the brain that are the originators of all movement or action or imagination. And that's sort of what Dennis was alluding to when he talked about the homunculus, the little man inside the brain. The little guy who sits up there at a high level interpreting all the brain activity. We can't do that. Somehow motor planning, when we make plans, it's not from activity at one little part in the brain, it's a part of integrated experience, bringing to it the sum of who we are, of our physiological state at the moment, whether we're hungry or not, of our memories of past events, of aspirations. I mean, at the highest levels of beauty, I imagine some of those architectural designs you had were far more beautiful than anything you could or have created. I mean, you were inspired by this vision of beauty, and all of that stuff works together to bring these plans into existence. So it's not one center in the brain, it's the totality of sort of who we are and what our experience has been and things we're not even aware of.
But is it worth also highlighting that the brain never sleeps properly, does it? I mean, it's always active.
Well, the brain's always active, even in sleep.
Yeah, exactly. Yes.
Yeah. So the brain is very, very active. Some of you have probably heard. I was telling someone that I was at dinner last night. I guess we were talking about this at one end of the table. Someone was asking me, is it true, this thing you hear about, you're never using more than 10% of your brain at any one time? And the brain is always active. And I was telling people, when you put people in an imaging, in a scanner, and ask them to do something like this for the first time, the cerebral cortex is wildly active, almost over the entire cerebral cortex. But by the 10th time you put them in a scanner and have them doing this, and this is they become accustomed to it and can do it almost without thinking about it, brain activity restricts, cortical activity restricts to a few loci, as though you're training up efficient networks to do this. And when you first start trying to learn something, anybody who's started learning a musical instrument knows this, right? You really have to think hard, hit the right keys or pluck the right strings on the guitar, and your brain is just massively active. But later, you can do some of these things by second nature. You train up subroutines, in a sense, and it frees up parts of the cortex to be doing other things. So the brain is always extremely active. I mean, it's your major source of expenditure of your energy resources. And the brain's fabulously expensive in terms of ATP, this little energetic molecule that is I'm babbling.
Yes.
I'd like to take up the question of consciousness, subjective experience being an emergent phenomenon, and I wonder whether we're actually undervaluing subjective experience by calling it an emergent phenomenon, because the word is used for, in other contexts, for things that we can measure that are in fact objective, for the example you gave, and the ice and water and so on. Whereas the subjective experience is something of a different order. And that if we allow it to be called an emergent phenomenon of the brain, then what happens to it when we die? Without the brain, we've kind of linked it less strongly to physical activity, which religion is absolutely related. But our religious experience becomes hard to hold on to.
So here's my belief about this, and it's strictly belief and strictly speculation. Okay, as far as I kind of think about this metaphorically. in a sense. So I think about a tune, or like Bach's air on the G string. I try to think about what is air on the G string. And air on the G string ultimately is a relationship of notes. It can be instantiated on a piano, it can be played on a violin, it can be played on the guitar, it can be recorded on sheet music and continue to exist even when no one in the world is playing it on any instrument at a given time. It is an entity that is real, has ontological status to me, and can be played out on a number of physical instruments. If all of those physical records were ever lost, then air on the G string would cease to exist unless it exists somewhere in the mind of God. If all the sheet music were lost, if no one alive had it in their brain at the time, it would go away. So, but my belief is that our selves, in a sense, and our consciousness, in a sense, is very similar to that. It's instantiated in this systems of neurons in this body that we have right now. And just as air on the G string can go from a piano to a violin, I see no logical problem with what's really me going from this body to another instantiation that might survive this body here. So there's nothing logically wrong with that. And I think John is probably going to address this in one of his talks later about life after death. So I would say, if God can take, this cumulative experience that's in my nervous system and transform that into a different instantiation, there's nothing illogical about that. We will continue to exist. I like the music metaphor because we all know that we can take a particular piece of music and transform it from a minor key into a major key. Okay, so if I have an existence beyond this life, I don't want it to be exactly the way I am now, because there's too much brokenness in the way I am now. There's too much evil in the way I am now. And I, like Paul, look forward to the new creation. So something that's recognizably me, but transforms me from that minor key into a major key is what I would like best. So I do believe that consciousness is an emergent property of our nervous systems, but I don't think that necessarily chains it or ties it inextricably to our nervous systems, that it can be transformed in a different way, and that's a part of our faith.
I'm a little concerned about the language, because people talk about it's just an emergent phenomenon. And then it becomes very devalued, and it is of a different order.
It is a different order. I mean, there are lots of different emergent properties. Attention and memory are emergent properties of the nervous system, but they're not as mysterious. Memory is, what the guys call the easy problem of consciousness, because I can describe memory in the third person. We can create memory devices over here. So you're right. I think consciousness is different from memory because it may involve this basic category distinction about the first person versus the third person.
Absolutely, and with everything you've said, the other example is a computer program which can be moved from one instrument to another.
Absolutely.
The personality
Absolutely.
still goes with the rather like a computer program.
Yeah, but perhaps the computer program can actually be improved in its future instantiation.
And that's a machine that's not about to crash.
I think it's key to avoid the word just and nothing but, and these sort of put down words.
Yes, exactly. Exactly. No, I would never say just an emergent property. I mean,
good. I think Bill's already spoken to us about the cookies waiting downstairs and that kind of thing, and I think we needed that. But I think we really want to thank Bill for a great talk.